The Regeneron Genetics Center, or the RGC as we call it, is our genetics discovery unit. We apply the best and latest technologies in sequencing and analytics to harness the power of human genetics to create game-changing new medicines. What we do differently at the RGC is application of technology and data at a scale that hasn't really been approached beforehand. We literally are pushing these technologies and the data to their limits, and studying thousands of diseases really allows us to systematically look for those big genetic drivers, those big clues and discoveries, wherever they may be.
We do have a really, really large and incredibly gratifying data set to work with. There are not that many institutions in the world that have invested the time and effort that we have to build a heterogeneous data set that can speak to a lot of the questions that we have and that the community has about understanding better genetics to help inform biology and to help inform the drug discovery process. Our perspective on AI and ML at the RGC is that it's really about the data. So the technology is there for having computers really learn meaningful patterns from data and applying those tools to then extracting insights from the data. But the computer has to learn too. Fundamentally, you have to have the data to train the model to understand the data. We try to build large, valuable, and really informative data sets so that we can, as individuals, extract the insights from the data.
The 1 million Exome project is the world's largest catalog of genetic variation, encompassing diverse ancestries. This dataset was made possible because we have sequenced more than 1 million individuals, and all of the sequencing has been done in one center. So this provided us an opportunity to make a harmonized dataset across various cohorts.
There are a number of benefits of increasing genetic diversity in genetic studies. Diversity in genomics offers unprecedented opportunities for new discoveries of genetic variants contributing to health outcomes and health disparities, particularly in underrepresented minority populations. In fact, recent discoveries in minority populations have led to new therapeutics. An example of this is PCSK9 for treating high cholesterol, and this was identified in African Americans, but this therapeutic actually benefits all populations. So there are a lot of opportunities here, and increasing genetic diversity really can provide new insight into health outcomes and health disparities.
So we've been trying to understand the host genetic factors that determines who gets infected with the SARS-CoV-2 virus, and for those who are infected with the virus, who goes on to get seriously ill or die. The gene ACE2 actually encodes the human host receptor for the SARS-CoV-2 virus, and we found that a genetic variant just upstream of ACE2 actually modulates the expression of this protein and contributes to who gets infected with SARS-CoV-2, and for those who are infected who go on to get more severe disease. So the impact of this research tells us that if we could modulate the expression levels of ACE2, we might have an impact on susceptibility to SARS-CoV-2 virus, or more broadly to other classes of coronaviruses.
GPR75 is a G protein couple receptor which is expressed in the brain across species. Earlier this year, we reported in Science magazine that individuals who carry rare mutations that truncate a copy of the GPR75 gene are associated with lower body weight and substantial protection against obesity. Approximately 1 in 3,000 people that we've sequenced in that study carried one of these protein truncating variants, and individuals with those variants had approximately 5 kilograms lower body weight and half the risk of obesity compared to individuals without those mutations. So we're really hoping that this genetic discovery may pave the way to eventually help develop medicines that can help people who struggle to maintain a healthy weight.
There's so much great science going on around us that it's kind of infectious. It's hard not to just get really excited about the opportunities to learn from the data sets and from the research projects we have, because there's just so much going on. And I think is the most exciting place in the world to be, because we make so much data with so many partners, and been really lucky, frankly, to be able to work with so many of the greats in the field and so many scientists who are just doing really amazing work all across the full spectrum of what happens in biopharma and biotechnology.
One thing I can tell you for certain is that genetics and genomic technologies are the absolute future of drug discovery and development. We are in a golden era of genomics technologies, which include RNA interference, CRISPR and gene editing, mRNA-based therapeutics and vaccines. We now have an avalanche coming of these new technologies, and they're absolutely increasing our ability to go after the best targets and to do it as fast as humanly possible. In parallel, we've also unleashed these genetics studies, genomics technologies, in all the types of work that we've mentioned. And we are so rapidly uncovering the genes that are important for disease, and that we can now go after and point these new technologies to hopefully cure many of these diseases.